Today, I'm going to give some background information that's critical to understanding the concept and the discipline of evolutionary developmental biology. And what I'm going to be doing here is I'm going to be presenting to you with my camera, and I'm going to be annotating the lecture slides on my tablet computer. So you're going to be seeing me look down quite a bit. So I hope you don't mind looking at the top of my head very much. So as we go, I'll be drawing on the slides and you'll see my annotations show up in this video. Here we have an image of a bat and a flying squirrel, both of which can fly. They're both mammals, but they're not closely related, which means that these winged structures, or their ability to fly, isn't the same. It doesn't have the same genetic basis. It doesn't have the same developmental basis. For example, the bat's wings are essentially modified fingers and webbing in between them, while on the other hand, the flying squirrel has webbing between its limbs. So in a sense, this is the ability of development to produce the same sorts of traits or abilities, but using different developmental mechanisms. That is, the cells and the types of cells that are in the flaps of skin that let this, actually, is that a flying squirrel or is it a sugar glider? My apologies to the mammalogists who are watching and who now don't believe that I know what I'm talking about. But regardless, this developmental trait is different from this developmental trait. The cells that are in between the fingers, so to speak, are different from the origin of the cells that are in between the limbs. So a big question in developmental biology then is, what is the genetic origin of novelty? How do these sorts of traits evolve? And what sorts of DNA sequence changes produce them? So to do that, it's important to understand, to revisit if necessary, the central dogma. The idea that each of our cells has double-stranded nucleic acid that we call DNA, and that encodes the instructions for producing ribonucleic acids, or RNA molecules, that are single-stranded, usually, versions of a nucleic acid, and that those in turn produce proteins. And these are structures, molecules, that actually do the work of the cell. These are the molecules that do DNA transcription and translation and provide cellular structure and build cells and send signals to other cells. So sometimes we'll diagram proteins as having a three-dimensional structure. They're physical objects. Some of them are structural proteins, some of them are enzymes, that is, proteins that catalyze reactions that actually change chemical structures inside of cells, and they perform other tasks as well. So that first step is called transcription, and the second step is translation. So DNA encodes RNA, encodes protein, the central dogma. And the reason this is important is because proteins, being the molecules that do the work of the cells, are the proteins, they're the objects that actually cause developmental change. And mutations in the DNA sequence then cause changes in the RNA sequence, which can cause changes in the protein sequence. So it's mutation of DNA that can cause proteins to change, and those in turn can affect how the cell operates. And an important and critical type of protein or class of protein that we're going to talk about a lot in this class is called a transcription factor. Let's forget that or enhancer bit. Let's just call them transcription factors. Enhancers are actually DNA sequences. My mistake. So these are a protein. They're produced from a gene. And what they do is they regulate what other genes are transcribed. So these are regulators of what genes get read inside the cell, and therefore what proteins each cell type produces. And it's the cells and the proteins that they produce that dictate back to the cell what sort of a cell that cell becomes. So the DNA encodes proteins. Proteins inform the cell what sort of a cell it should become. It can make cells skinny and flat like epidermal cells. It could make cells long and skinny like neurons. Now, the proteins are the things that are doing that work, that make cells that have the same DNA sequences. All of the cells in my body have the same DNA sequence. What makes them different from each other is that each of my cell types produces a different set of proteins. And it's these transcription factors, proteins themselves, regulate the production of other proteins. So the set of transcription factors that are present in each cell will differ, and that's what causes cellular differentiation. And the way that transcription factors work is they tend to bind DNA to regulate which genes, which stretches of DNA that encode proteins get turned on or turned off. So here's an example transcription factor, and this is a general transcription factor, or GTF, which means that it is a transcription factor that operates whenever transcription occurs. That's why it's called a general transcription factor. There are other transcription factors that are more specific, but this protein, the TATA binding protein, which is also usually abbreviated TBP, is one of the first proteins that interacts with DNA at the start of transcription, the first step of the central dogma. So what it does is, this is a cartoon of the protein structure of TBP. It recognizes the DNA sequence TATA, hence its clever name, the TATA binding protein, and it will bind to DNA where it sees that sequence. It's actually a longer sequence than just those 4 nucleotides, but those are the core essential nucleotides for TBP to bind to DNA. And when it binds to DNA, when it recognizes that sequence and the protein approaches the DNA double helix and physically binds to it, chemically binds to it, it starts recruiting other proteins, other transcription factors like TF2A and TF2B. Don't need to memorize all of these names. And that set of proteins together produces a stable complex that recruits RNA polymerase II, which is the protein that actually does transcription. It reads the DNA and produces an RNA molecule. So TBP is an essential component of transcription in all of our cells. If we didn't have TBP, we would be not alive. Here's another view of TBP. In this case, this is the fission yeast Schizosaccharomyces pombe TATA binding protein bound to DNA. And you can see that its physical structure allows it to interdigitate these alpha helices, part of the structure of the protein, where amino acids that are the components of a protein form spirals by chemically binding with each other. And it's these contacts between the spirals and the hydrogen bonds in the double helix of DNA that tell TBP when it's found a TATA sequence. In other words, the interaction between proteins and DNA is a chemical interaction, and those chemical bonds between the protein and the DNA help this protein, TBP, stick to specific DNA sequences when the hydrogen bonds that can be formed between the amino acids in the protein and the hydrogen bond acceptors and donors in the DNA are able to make those bonds. And those chemical bonds hold the protein and the DNA together, not permanently, transiently instead. But the better the fit between the protein sequence and the DNA sequence, maybe the longer that this protein, TBP, will stay near the DNA. And the longer TBP stays attached chemically to the DNA, the more likely it is to be able to recruit the other transcription factors and eventually RNA polymerase and initiate transcription of a gene. And then I've mentioned that transcription factors can cause the production of other transcription factors. These proteins then, by binding to DNA, can either turn on transcription of genes or turn off transcription of genes. And this can be problematic but it's essential for life. So here's how this works. You can consider one protein, like a gene and a protein we'll call HOX1, that's expressed in the anterior of a developing organism. So assume that this gray oval is a multicellular organism that's an embryo at this stage. So there are a number of cells in this red region that express or have this protein HOX1. We'll use the word express a lot when we're talking about the central dogma and genes. We'll talk about genes being expressed. And what that means is basically that the central dogma is occurring to that gene. So when a gene is expressed, that suggests that it's being transcribed into RNA, and the RNA is being translated into protein, and that protein is present. So that gene is essentially expressing its presence in that cell. So all genes in the DNA are present in every cell. The question in development is which genes are expressed, turned on. Are there proteins, or sometimes just the messenger RNAs themselves, present in the cell? That's expression, either the presence of the messenger RNA or the protein, or both. So in this example, HOX1 is expressed in a certain set of cells, and this gene and protein that it produces, HOX1, positively interacts, and that's what the arrowhead indicates, is the HOX1 protein turns on expression of this next gene, I1. And let's say that produces a blue pigment in the organism. So we can see by looking at the organism that there's a region here where the I1 gene is being expressed, its protein is being produced, and that protein somehow, magically in this example, is causing these cells to produce a blue pigment. And then let's assume that HOX1 can also positively regulate, that is, promote the transcription or expression of another gene, EAR1, that produces the protein EAR1. And that produces also a yellow pigment. So we can see its region of expression here in the anterior or head end of a developing embryo. So here's the question. If HOX1 positively regulates I1, and that's supposed to be produced where the eye of the developing organism will be built, and it also turns on this EAR1 protein, which is going to be regulated and expressed in cells that produce ears, we don't want both of those proteins on in the same cells, because we don't need cells that are trying to figure out, am I supposed to be eye, am I supposed to be ear, like these cells in the middle of this diagram. So how does that work? Well, it turns out, and this is again important just for understanding how these gene regulation diagrams work, is the flat-headed arrows indicate negative interactions between proteins, which we'll also call repression. So the positive interactions I'll refer to as turning on, but also they are activating. In other words, HOX1 activates I1, HOX1 activates EAR1, but those two proteins repress each other. So in a cell, if there's more EAR1 protein, it will act to turn off expression of I1. In other words, perhaps this EAR1 protein is itself a transcription factor, and maybe so is I1. So I'm going to write that one in blue, actually, to correspond with the coloring in this figure. So imagine these are both transcription factors. A transcription factor, HOX1, has produced the expression of more transcription factors, but in different cell types. So that we have some I1 transcription factor here and some EAR1 transcription factor here. And what those proteins will go on to do is to affect the expression or repression of yet other genes downstream in this cascade of transcription and translation, or the central dogma. So the last point I want to make on this slide is that there has to be some sort of a boundary set up here. I mentioned a few minutes ago, we don't want cells that are confused about what sort of tissue they're supposed to produce. We don't want cells that are both eye and ear cells at the same time. And that's why these repressive interactions are important, because what that will do is we'll set up a solid boundary in the tissues, the cells of the organism, where some cells will either have a little bit more blue than yellow, and they will commit to becoming blue type cells because there's enough blue protein that it shuts off the transcription and translation of yellow through the action of its transcription factor. And likewise, if cells are more yellow expressing, there's more yellow protein than blue, this balance will tip in the favor of whichever protein has more than the other. So if there's more yellow, it will more strongly turn off blue than blue proteins would be turning off the expression of yellow genes. And that sets up, in most cases, a strong boundary. And that's how different developmental tissues are defined. So I've already described this concept then of a regulatory cascade. You can have a central protein like a Hox gene, which we're going to be talking about more in just a little bit, that influences the expression of yet other genes, and they then can provide the specification for cells to turn into specific types of cells. And those can be both activating arrowheads and also repressing the blunt arrows. So HOX1A expression, the protein, will turn on the expression of the genes that are in green, and it will also turn off the expression of genes in red. So these sorts of figures we'll see a few times when we're studying evolutionary developmental biology. Now, to this point, we've only been talking about static processes, imagining in one cell at one point in time, there's a certain amount of yellow produced and a certain amount of blue produced, eye and ear. But we also have to think about what's happening over time, because development isn't a static process, it's a dynamic process that occurs over time. And cells divide and divide, and at some point in time, they have to shift or modulate what sort of cell they're about to become. So another thing that we have to consider is the stability of the different parts of the central dogma, transcription and translation. So the lifetime, in other words, of when a transcription factor turns on transcription of a gene, production of an RNA, that's happening all the time because the life cycle, or the lifetime of RNA molecules is pretty short. Once transcription happens and an RNA is produced, that RNA survives for minutes, seconds, hours, not very long in terms of the lifetime of the developing organism potentially. So this is one way that tissues are controlled, and that organisms essentially, or their DNA, controls what sort of cells can exist and how quickly the cell types can change as development progresses by changing the production of RNA. In other words, transcription factors, these proteins, will bind to DNA and turn on transcription, but not forever. The transcription will occur for a brief pulse, perhaps, and produce a small number of RNAs. And the lifetime, the amount of time that those RNA molecules exist before they get destroyed inside the cell, is short. So that means you can only produce a small amount of protein from a little bit of transcription. So transcription factor binds, produces a small number of RNAs. They're quickly translated by ribosomes, but then those RNAs get destroyed. And so then the ribosomes don't have anything to translate. So they can't produce any more protein. In other words, the protein content of a cell can change quickly over its lifetime. And that's important for developmental biology. Proteins themselves are longer lived generally than RNAs, but they themselves have processes inside the cell that can destroy proteins. So proteins are built from RNAs. They're encoded by RNAs. They're built by ribosomes. And then the protein is turned over or destroyed at some point in the future, and its amino acid components recycled and used to build other proteins. The process is dynamic. In a cell, over time, the proteins, the transcription factors will change. That will change the parts of the DNA sequence that are transcribed into RNAs and which proteins are produced in that cell. So cells aren't static. They don't always produce exactly the same proteins for their entire life. Another thing we have to think about in terms of understanding how development is dynamic at the cellular level is that the amount of protein can affect transcription. So if we're talking about proteins that are transcription factors that turn on the central dogma, their concentration matters. So for example, it could be that there's a little bit of transcription and translation of, say, this example protein transcription factor. So it will bind to the DNA where it recognizes a specific DNA sequence, like TATA binding protein recognizes TATA. And so it binding will cause transcription and translation, and maybe that's sufficient to produce whatever proteins it regulates. But what if you have more of that same transcription factor in the cell? With more transcription factor present in the cell, it's more likely by chance that one of those protein molecules has actually found and bound to the DNA sequence it recognizes and turned on transcription. So this chart at the bottom then is meant to indicate that when you have a small amount of protein, you're not going to have the protein bind to DNA, its target sequence, as frequently as if you have a lot more protein, the same protein, multiple different individual molecules of the same protein, product of the same gene. The more you have, the more likely it is that one of those by chance ran into physically the DNA sequence it recognizes, binds, and then turns on transcription and translation of that gene. So the temporal dynamics also not only includes what we talked about just a moment ago, but also we have to consider how much of the transcription factor is present at the time. And the degradation of that protein, the rate of its degradation, will also influence transcription and therefore what proteins are produced in the cell and therefore development. I hasten to add that there's also a role for the environment. And I don't necessarily mean, although it can include the actual environment of the outdoors, temperature, rainfall, humidity, pH of the environment, and so forth. But also, each cell doesn't act independently. And there are signaling processes by which cells in the neighborhood or even far away in the same organism can send signals to other cells that then influence the ability of transcription and translation, gene expression, to occur and to be regulated. So each cell itself is not necessarily what we call autonomous. That is, each cell doesn't act as its own little fiefdom, its own little kingdom or entity that ignores everything that's going on around it and does its own thing. If that was true, actually, development wouldn't occur because, as we've discussed, each cell in an organism has the same DNA. So each cell has the capability of producing the same sets of proteins. That's the concept of stem cells. Stem cells are cells that are so young in terms of developmental age that they still have the ability to decide which set of proteins to produce and therefore what type of cell to become. So cells aren't usually autonomous. Stem cells can be somewhat, but in this case, cells also take into account physical interactions and also chemical interactions, as in the case of hormone signaling, with other cells in their environment, in their neighborhood, even in the context of the body of the organism they're in. So I'll let you read this. I don't like text-heavy slides, but I thought this was a good way of making the very important point that it's complicated. And this is scary because that means this process, the central dogma and then development, is super dynamic. There are so many inputs that it can be hard to predict what's going to happen. And that's what we're going to talk about a little bit in terms of the rest of the course, is how can developmental biologists make any sense of any of this at all? So let's talk about development and evolution. So how does the expression of a developmental gene change? Here's an example of an apterous mutant. I think I mentioned this previously, but if not, this is a Drosophila melanogaster, a fruit fly, that has a mutation in one gene, apterous, and that gene is important for producing wings. So when you mutate the gene, when its DNA sequence changes such that the protein it produces can't do its normal job of building wings, you get a fly that shows the mutant phenotype, which means that it doesn't have wings. And so one of the questions is, how would you cause developmental change? How could you cause, for example, a fruit fly to lose its wings? Based on the central dogma, there are a few different major options. There may be more insignificant categories, but these are the major three. The part of the gene that encodes the apterous protein could change. So it changes the shape of the protein so that that protein no longer has the ability to create wings. Or you could mutate the part of the gene that isn't the part that encodes and produces the protein, but the part that transcription factors bind to that turns on the production of that gene, transcript and translation, the RNA and the protein in the first place. Or a third option is you could get an entirely new gene from scratch. So not a mutation to an existing gene. That would be one of the first two options. So if there's an existing gene, you can cause developmental change by tinkering with that gene. Or you could produce a new gene that has a new function that changes something for the first time in an organism. So these are three options in the context of this specific example. You could mutate the part of the gene that encodes protein. You can change the part of the gene that causes that gene to be expressed at all. Or you could get a duplicate apterous gene that has the ability to do a new thing. So for the rest of this presentation, I'm going to talk about Hox genes in specific, and then also how gene duplication can influence evolutionary developmental biology. The Hox genes are a set of genes that are essential for development in practically every organism that does development. They are a set of transcription factors, and the role of these individual genes and the proteins that they produce, these are examples from Drosophila, from Norton et al. Textbook. These proteins and the genes that encode them are found in one part of one double helix, one chromosome in many organisms. So this is a representation of a DNA double helix. And these proteins are expressed in the body of the organism in the same order in which the genes are found on the DNA, which is really striking and sort of odd in terms of genetics. It's not often the case that physical position of genes indicates anything about the order of expression of those proteins in the body of an adult organism. So they're kind of a cool gene family in themselves. So they're usually found near each other in clusters. So in this example, there are 8 different Hox genes. And Hox, again, stands for homeobox. It's an abbreviation of homeobox. Apterous is one of them. And it's the cells that produce each of these cell types that turn into those particular segments. So antennapedia, ANTP, for example, is a protein that's produced that's important for thoracic development. Ultrabithorax, UBX, is another gene that's also a transcription factor, but it's turned on and is important for cells that are produced and cell types that are produced more towards the anterior of the thorax. What makes a Hox gene is they all have a particular DNA binding domain called a homeobox. What this means is the gene that encodes a Hox protein has a particular type of DNA sequence that when it's transcribed and translated and turns into a protein, that protein has a particular shape As with TBP and its interaction with the Tada box that we looked at earlier, that shape helps it bind specific DNA sequences. Hox genes, all of them, because as we just saw, there are many different Hox genes, usually found near each other on a chromosome. Those many Hox genes exist and are broadly conserved among species. This harkens back to this concept that the purpose of development is the reproduction of very similar looking organisms, members of a species is one way to think about it, from the same genetic code. So development needs to be robust against change. And that predicts that basic essential developmental genes should basically have the same DNA sequence and the same protein structure and the same function among even very diverse organisms. Because the initial stages of development in all mammals, in all animals, are very similar. We all start out as a single cell. That cell duplicates and becomes 2 cells. Those 2 cells duplicate, divide, and become 4 cells, and so on. So early development is highly conserved. The way that we get novelty is usually in changes in later development. And one of the reasons for this is because changing early development could really mess up all of the steps in development that happen after that intervention. And the later development has already progressed, the less likely an organism is that a chance mutation that causes a change screws everything up. Maybe it changes one thing, like the shape of a wing or the slight color of a wing, or maybe the length of a wing in case of apterous. So in this case, this is a demonstration that Hox genes are broadly conserved. You can get the apterous mutant in Drosophila that doesn't have wings because it's missing the gene that's necessary to produce them, this Hox gene apterous. But the Hox genes are broadly enough conserved that you could take the same gene, and this is an important term, an ortholog. It means the same gene, protein, or function, but different species. So if you're talking about the same gene, like apterous, it's called apterous in Drosophila, but humans have the same gene that has the same function. It doesn't make us grow wings, but it has the same basic developmental role in humans. And this ortholog, you can take out of a human, you can get human DNA, the same gene, the orthologous gene, and put it in the DNA of Drosophila apterous mutants that normally look like this. And lo and behold, that human gene encodes the apterous protein, the orthologous protein, and that protein, even though it's a human protein, works in the exact same way, almost, in Drosophila to allow that mutant to once again grow its own wings. So that's an expectation of development, that developmental processes, especially basic ones, resist change. When mutations happen in essential developmental genes, usually really bad things happen. And that's why the answer to evo-devo and how development changes is not as simple as, so you just mutate your Hox genes, right? Absolutely not. Look at what happens with an ultra-bithorax mutant. So you get rid of this protein, and all of a sudden, the Drosophila has 2 thoraxes, bithorax, and 2 sets of wings. Yes, this does produce novelty. So this is a form of evolution, and that's the developmental basis for evolution in this case, a duplication, or a mutation rather, in a gene that duplicates a body segment. That's a great way to change. Then the evolution part of the developmental question in evo-devo is this a good change or a bad change? Depends. Is this fruit fly going to be able to find a mate? Is another, assuming this is a male, I can't tell. Let's ask ourselves, do you think a female fruit fly of the same species would recognize this individual as a member of her species? If she did, would she allow the male to court her? Would they successfully mate? Would they have offspring? I don't know. But if they didn't, then this would be a deleterious or a bad mutation. you've had novelty, but unless that mutation is fit in the organism it produces, then it doesn't get passed on in the DNA code of this individual to its next generation. So there's no evolution. If this mutant is the only mutant and it's not healthy, if it's not fit, if it's not alive and able to produce offspring of its own to pass that mutation on to the next generation, that is not evolution. So evolution of development doesn't necessarily involve major changes all at once. Usually minor, small changes happen. That's a way for novelty to be tested in the natural world. In other words, evolution. For example, that previous example of ultrabithorax mutant has a big consequence on the fly. It could be a really beneficial consequence, or it could be a very negative consequence. So what's been discovered over time is that developmental changes tend to take small baby steps, small changes at a time. And so if that change increases the fitness, the health, the ability to reproduce of an organism, maybe it has a chance of sticking around longer in the subsequent generations. And eventually maybe that change will become typical in that species. And if that developmental change is a little bit negative, that doesn't affect the health of an organism too much, then it can stick around into subsequent generations too. But if the offspring and the offspring's offspring and the great-grandchildren and the great-great-grandchildren aren't as healthy as organisms that don't have that slightly bad mutation, over long periods of time, over generations and generations, that slightly deleterious or bad mutation will disappear. It'll be replaced by the organisms that don't have it. So let's take a look at one example of how these sort of subtle changes can occur. In this case, we're looking at two different binding sites for transcription factors for one gene on a chromosome. So this is an example of the yellow gene in Drosophila. And it's involved in transcription in a particular area of the wing. And it's also involved in in production of pigment in the abdomen. And so what this is showing is we're looking at this transcription factor, which can bind to DNA and turn on expression of the yellow gene. And we're also talking about this transcription factor and its ability also to turn on the yellow gene. So normally these transcription factors will bind to DNA at their binding sites, and they'll either turn off or turn on production of pigmentation. So here's an example of a mutation that doesn't occur in the protein coding part of the gene, which is here. That's the protein coding region, the part of the gene that encodes RNA that will encode protein. Instead, what's happening here in this cartoon example is the situation where perhaps a small DNA change occurs that changes the DNA sequence and allows a new transcription factor to bind. And what that binding does is it turns on the yellow gene. And if that transcription factor is expressed, if that protein is only being produced for other reasons in a small area of the wing, that will turn on yellow protein production in that part of the wing. And in this case, again, the yellow gene produces a protein, the protein called yellow, that's involved in the production of melanic pigments, dark pigments. So in the original case, there's no black spot on the wing because the transcription factor doesn't bind there. Mutation causes a change in the DNA sequence to allow this transcription factor that was present in the cell already, but didn't have a place in the DNA to bind, gives it a sequence that it recognizes. And because that protein in all of the cells of the organism is only present in the small patch in the wing, this transcription factor only in that wing spot area can bind to the DNA and produce yellow protein, and that produces pigmented wings. So instead of a fly with no wing spots, you get a fly with wing spots. I do want to reiterate, because this is important, the reason this happens is because, for reasons we haven't discussed, that red transcription factor, the protein, is only being produced in those tiny regions of the wing already. What that means is that the gene that encodes that transcription factor is only being transcribed and translated in that little area of the wing. So this is sort of opportunistic. The protein, this red transcription factor, was already in these cells. The reason it wasn't able to produce protein that causes pigmentation is because it did not have a binding site near the yellow gene. Once mutation caused it to have a binding site, then where that protein is present in those wing spots, it then could turn on yellow production, and that's why pigment shows up. And the opposite can happen as well. You can lose a binding site through mutation. So in this case, we're looking at the example where this transcription factor, which is important for the pigmentation of the abdomen, a DNA sequence change occurs here just upstream of the yellow gene, and that sequence change removes the DNA sequence that that transcription factor initially bound. So it can no longer turn on yellow expression. So you get a Drosophila that did have a pigmented abdomen now no longer does. So those are two examples. One, a DNA sequence change, a mutation that creates a transcription factor binding site, and that being a cause of developmental change. And also a mutation that causes the loss of a transcription factor binding site, and that causes another developmental change. And the important point again here is that these are minor changes. They're not body-wide. They're restricted to particular areas of the body, and they make subtle changes, as in this case, pigmentation. Probably not something that if pigmentation screws up in development, that the organism is dead. But it maybe does change its fitness. It maybe does change the ability, as in the previous cartoony example, of will a female recognize this individual as a member of her own species or not, because the wings look slightly different or because the abdomen isn't pigmented the same way that she expects. So these subtle mutations that make small changes can still have big consequences. Now, the next type of evo-devo is not by these subtle changes in gene expression, but you can also get developmental change by gene duplication. And what I mean by that is the Hox genes have changed over time. So the Hox genes are a great example of this. Invertebrates don't have Hox genes. And the lancelets, cephalochordates, have one cluster of Hox genes. So this is where Hox genes originated, essentially, the ancestor of vertebrates. And these basal or ancestral organisms that still exist today just have one set of Hox genes. Over time, that one set or cluster of Hox genes duplicated. So the whole part of the chromosome that contained all of those Hox genes basically copy and pasted itself somewhere else on a different chromosome in the genome of these organisms, such that jawless fishes have at least 3 sets of those same Hox genes, 3 clusters. Chondrichthyes have 4, sharks and other cartilaginous fishes. And then in ray-finned or bony fishes, they had a whole genome duplication occur so that the number of Hox gene clusters in this specific lineage down here, close to present day, the set of Hox gene clusters duplicated. So we went from 4 to 8, but just in that set of organisms. In most other vertebrates, including us, tetrapods, mammals included, have 4 sets of Hox genes. So in the cartoon I showed from Drosophila, there are several Hox genes all in a row in one region of the chromosome. We, humans, have 4 independent sets of those same Hox genes. And that is really important. And this is thought to be one of the major ways that evolutionary development happens. Not necessarily, although it does happen, using those individual mutations perhaps to transcription factor binding sites like we saw. But the production of duplicate genes provides the basis for immense change to development in a safe way. And that's what we're about to discuss. So again, Hox genes are broadly conserved among species. They tend to be, because they are all inherited from the ancestor to all vertebrates, that part of the chromosome tends to be the same. It's got the same genes here colored and also named by their fruit fly nomenclature, like Antennapedia. But look at the color codes. The fly has 1, 2, 3, 4, 5, 6, 7, 8 shown here in this cluster. There are more. And if you look in mice, remember we just mentioned that there are 4 sets of clusters, or the 4 clusters rather. The cluster 1, cluster 2, cluster 3, and cluster 4. They're on different parts of different chromosomes in the genome of the mouse. But look at the genes. This is a fly gene. It has 1, 2, 3 orthologs, remember, the same gene in different species. So those are orthologs. I'm making this point because we're going to use this terminology a lot, orthologs. Notice that one of those orthologs is missing on the 3rd of the 4 clusters in mice. So this gene has 3 orthologs in mice. Those genes that are the same, but there are multiple copies in the same organism, are called paralogs. So orthologs is the word we use to define the same gene in different species. If there are multiple copies of the same gene in different regions of the chromosome, we refer to those as paralogs. So in other words, HOX1, this version, the cluster 1, is an ortholog to the fly version. The HOX2 cluster is also an ortholog. The HOX4 is also an ortholog. So between species, we call them orthologs. Within a species, these 3 are paralogs of each other. 1 and 2 are paralogs, 1 and 4 are paralogs, and 2 and 4 are paralogs. One of the important points here is to note that, as I just pointed out with this, there's an ortholog missing on the HOX3 cluster. There are also some others missing in this cluster, and then some of the clusters have new versions that aren't present in Drosophila. So there's a lot of variation in the presence of these paralogous genes within vertebrates. Each of the 4 HOX clusters doesn't look exactly the same in terms of the genes that are present. And to complete this slide, I will also point out that these genes tend to have not only the same gene order on the chromosome, but also have the same expression pattern. That the genes at the 5' end on the DNA tend to be expressed anteriorly, and then posterior expression corresponding to the 3' end of the cluster. So this all fulfills that prediction of development, that basic developmental genes like these genes that cause basic anterior-posterior or head-to-tail patterning, which if you screw it up would be really bad for the organism, are really strongly conserved among organisms. So the end of this slide makes a really important point that I'm about to discuss in detail, which is what's the benefit of having all these duplicate genes? That's where we're headed. So development can cause changes by making subtle changes or making big changes with gene duplication. So here's an example of gene duplication. This is one of the ways that HOX gene clusters duplicate. It is true, although it's rare, that regions of chromosomes can break and copy and paste so that you get 2 adjacent or on non-adjacent, potentially, sets of the same genes. And this is great for evolution because those extra genes give the substrate for sort of tinkering around based on mutations. So this is another view of essentially the same cartoon showing different species this time, where we're looking at Drosophila. Here's the hypothetical ancestor. So the ancestor of all living organisms, well, insects and vertebrates at least, potentially had those 7 HOX genes. And this suggests that this HOX gene may be duplicated a few times in the lineage leading to Amphioxus. And from that ancestor to chordates, there were 2 duplications, and that's potentially why we've got 4 copies of something that looks very similar to this older lineage that has 14 different Hox genes. And then what we do is we can look in mice and in zebrafish. And again, remember, zebrafish have twice as many Hox clusters as mice because of the genome duplication that bony fish encountered. Look at the variation in the presence and absence of paralogs. Remember, each of these is paralog. So here's Hox1, 3 paralogs. So the same gene, but different copies, 3 different locations in the genome on different chromosomes, potentially. One of them's missing, so there are 3 paralogs. 2 paralogs of Hox2, 3 paralogs of Hox3. And this is different from zebrafish. So some of these duplications have led to mice, for example, still have all 4 copies of Hox9 they started with, but they're down to 2 copies of Hox7. On the other hand, fish at this point, at least zebrafish, only have 1 of the original 4 or 8 actually Hox7 genes that it started its lineage with. So not present day zebrafish, but the ancestor of the zebrafish. So here, we're already seeing diversity. We're seeing diversity in how many and which Hox genes are still present. And maybe that in itself explains the differences between these sorts of organisms. Maybe it's the case that having more or less of one of the Hox genes is what causes mice to look like mice and not zebrafish, and vice versa. So one thing that we'll discuss in a little bit is this raises the idea of a Hox code to development. That perhaps the set of Hox genes that you have as an organism dictates something about your development. So here's a particular example of a duplication. We've got a loss of 2 different Hox genes in mice. But so duplicate genes, what does duplication do to benefit development and evolution? So here's a great figure from a review from Evans in 2008 that sort of explains what the possible fates are of duplicate genes. So we start from the left. A gene duplicates. So we've got 2 different versions of a protein, let's say, that that gene produces. This is meant to represent a double helix, but we're going to think about it in terms of the functional part of the gene, which is the protein. So duplication, now we've got 2 versions of the same gene. If both copies of that gene are expressed, that is, if the 2 copies are both transcribed and translated and produce proteins, then there are 2 different things that can happen: neofunctionalization or subfunctionalization. And I'll give you some good analogies of this in just a minute. But neofunctionalization suggests that the original copy maintains its original role. That is, the duplication gives you an extra copy of a gene for evolution to play with. And in this case, neofunctionalization suggests that now that you've got the original and a copy, what would happen if a mutation happens in the gene that encodes the copy? You can evolve something new. That protein shape will change. Maybe that protein shape that's new in the copy does something new for the organism. That's new functionalization. So that's if a beneficial mutation occurs, something that's a helpful mutation to the organism. We would call that neofunctionalization. That new copy has a new job and it's useful for the organism, it's helpful. If not, if the mutation occurs, but it's not a beneficial mutation, then we ask the next question. Was it a bad mutation or was it a neutral mutation? So if a mutation occurs, but it's not beneficial and it's not detrimental, it's neutral, all you've got now is you've got 2 copies of the gene. That's called redundancy. You started with 1 copy of the gene, it could produce a protein. Now you've got 2 copies of the gene, the second copy can still produce the exact same protein. So this is neutral. It doesn't really do anything yet, good or bad for the organism. If a mutation occurs to the copy that is deleterious or negative, what that can lead to is subfunctionalization. It's a possibility. not sure, but one of the opportunities it has is that original protein maybe had two different jobs it did inside the cell. So when you've got two copies of the gene, if each copy sustains different mutations by chance, that means that one copy, copy one of the gene, one paralog, might get a mutation that disables its ability to bind to DNA, but it still has its enzymatic function to phosphorylate some other protein. And then the mutation in the other copy of the same gene might retain its ability to bind DNA, but it prevents it from doing its other job. It's not usually the case that a transcription factor would also be a kinase, but let's say it allows it to continue to do its job phosphorylating another protein. So that would be subfunctionalization. If the original gene has multiple, encodes a protein that has multiple different functions, if you've got two copies of this gene and produces two proteins, you can get different sets of mutations that happen in the two different copies that let those two proteins do different sets of the same original function. Subfunctionalization. And then if both copies of the duplicate gene aren't expressed, that means one of them got destroyed or otherwise was unable to be transcribed and translated, then that's non-functionalization. You're back to the same position you were at originally. The duplicate gene was created, it was destroyed, now we're back to square one. So it's neofunctionalization, subfunctionalization, and non-functionalization. Let me show you some, what I hope will be useful analogies for this. Neofunctionalization. You've got a phone. This is an old phone. I should probably update the graphics. And then all of a sudden you get a duplicate. Well, you still need that phone to do its original job, which is to let you talk to your friends. But now that you've got a second copy, you can use that for a new purpose. In this case, you could keep that original copy of the gene, the protein, or the cell phone for its original role. And now you can use your copy to be a doorstop if you need to. Right? That would be a new function of this protein. It's doing something good for you that you couldn't do before when you only had one, because you had to save it for its original function. Subfunctionalization would be something like taking a smartphone. It has two different roles. It's both a telephone and it's also a computer at the same time. So if you get duplicate genes, one of those genes could lose its ability to be the computer by mutation, but it still allows the organism to make phone calls. So it still allows the telephone function of the original gene or protein to work. And the copy of that gene might, by mutation, deleterious or negative mutations, lose the ability to make phone calls, but that still lets the protein act like a computer. So duplication leading to subfunctionalization means the organism with both of those copies of the gene and the proteins they produce can still function. They're still healthy because they can still do the original function of the organism. They just have to use two genes now to have the same result as what they could have done with the original protein. And then non-functionalization. You get a duplicate, one of them breaks. That's fine. You're still a healthy organism because you still have the ability to do the original functions of the original gene and protein. And in fact, that's a benefit of gene duplication, is they provide a backup copy. And that's how they benefit evolutionary developmental biology. Gene duplications gives a target, a gene, the copy of the gene, for mutation to act in and to try, sort of test out new mutations and see how they work in an organism. Do they benefit? Do they detriment it? So not that Mother Nature and evolution is forward thinking. They're not. All mutations happen randomly. But we tend to describe it as being this sort of forward-looking testing mutations. That's not actually what's happening. It's trial and error. But that is how evolution proceeds, is random mutations tinkering, so to speak, with genes and the proteins they encode. And then eventually, over time, we later discover whether or not those changes were more or less beneficial than what was already existing in the population, the original wild type sequence of the gene. So non-functionalization, this is what's happening when we see Hox clusters that are missing paralogs. These paralogs are here because they are all have some function. These could be subfunctionalized, they could be neofunctionalized, but the fact that they're all still here means they still have some function. The ones that are missing likely were non-functionalized. They got mutations that destroyed their ability to work, like that broken smartphone. And so they don't exist anymore. But that doesn't necessarily do something bad for the organism, because even though they're missing those paralogs, they still have 3 other copies of both of those genes. And then we've got redundancy. And that, again, is what I was just describing as the benefit of gene duplications. It gives you a backup copy. If one of these happened to break in the future because you drop it or because a mutation happens in the gene that it represents, you're still good because you've got a backup copy. So that's why gene redundancy might be good. And what I'd like you to think about and consider is, why might gene redundancy be bad? Is there any downside to having copies of genes? And I've already mentioned this before, but just to reiterate, in fruit flies, for example, the order of Hox genes corresponds with the position in the organism anterior to posterior that they're expressed. And this is true from fruit flies to mice. And this is, again, that concept of the Hox code, which proposes the idea that where the Hox genes are expressed tells those cell types what sorts of cells to become. So Hox5 is expressed in cervical tissues, both in the chick and also in a mouse. And where we see changes in the types of tissues, so there are more cervical vertebrae in a chicken than there are in a mouse. And where we see that developmental change, we also see a shift in gene expression. So where Hox5 isn't turned on, we don't see cervical vertebrae. We start to see Hox6 through Hox9 turn on, and those define the thoracic vertebrae. And they're expressed in the greater number of thoracic vertebrae in a mouse. So again, the Hox code, that there might be a specific Hox gene, one from each cluster, that is specific to a particular cell type in anterior-posterior head-to-tail patterning. So this point here is that comparative anatomy, the ability to look at gene expression and tissues in 2 different organisms, the comparative part, suggests that this is the case. There's one difficulty with this is the paralogs, because how many different Hox5 paralogs are there in a chicken and a mouse? And how would you know for sure which of the paralogs are important for this process? Is it all of them? Is it some of them? They probably have redundant jobs, because if they're still present, they may all be redundant. So how would you go about figuring out whether or not a gene that you think is important for development actually plays a role in development? And this is made more difficult, again, in this example, by the idea of paralogs. And I'll explain why that is in just a minute. So this is the last part of this presentation, and it's to answer that question. How do you study the function of a gene in development? So there are 2 basic types of experiments that evolutionary developmental biologists do to study gene function and whether or not a gene is either necessary to produce an outcome and if it's sufficient to produce an outcome. Necessity and sufficiency are really fundamental concepts in genetics in general, but especially in developmental biology and evo-devo as well. So here's the concept. If a gene is necessary to do something, like let's say that a Hox gene, HOXA1, for example, let's say it's necessary to produce, I don't know, let's come up with something weird, like extra legs. If that protein is necessary to produce extra legs, how would we prove that? And when we mean necessary, we're talking about it's essential. You have to have it to get extra legs. So how does a biologist test this? Lose it. So for example, what developmental biologists might do is delete the HOXA1 gene, find a mutant of that organism, or these days use CRISPR-Cas9 or some other method to remove that gene from the organism. And the prediction is, if HOXA1 is necessary to produce extra legs, and you get an organism that doesn't have that gene, if it's necessary or essential to produce legs, if yes, if it's necessary, then you get an organism that doesn't have any legs. And again, the problem with paralogs is that it could be that HOXA2, another one of its paralogs, has the exact same role as HOXA1 in the organism in the production of extra legs. So if you get rid of HOXA1, one of the paralogs, and you still see the production of extra legs, that means it's not necessary. So how many other HOXA paralogs do you have to get rid of before you find what set of those genes is necessary to produce extra legs? It's a complication in the study of developmental biology. In terms of sufficiency, that means that it's sufficient, it's enough, it's all you need. Or enough. That is, by itself, is HOXA1 necessary to produce, by itself, is HOXA1 all that you need, sufficient to produce extra legs? So what you would do in this case is what developmental biologists colloquially refer to as a move it experiment. You would put that gene, make it expressed, make that gene, HOXA1 in this case, transcribed and translated, expressed, in a new cell type that doesn't normally produce that protein. And if that's the only change you make to that either cell type or sometimes organism, you can move a gene from one cell type to another cell type to test sufficiency, or you could move one gene into a new organism, a different species that doesn't have that protein. If they still grow extra legs, then yes, that gene is all you need. It's sufficient to produce extra legs. If you take that gene and it's not sufficient for whatever trait you're studying, you put that gene and then the protein it produces after the central dogma into a new tissue type or new organism, and it doesn't have the effect that you're looking for, then you know you've just made one change. You've added a gene. And if that gene doesn't do anything, then it's not sufficient. It by itself is not enough to cause the development of that trait, that change. So we'll see these two terms repeatedly in the study of evolutionary developmental biology. Thanks for watching, and we'll look forward to seeing you on the next version of this video series.